The sliding filament theory. The sliding filament theory takes place in the muscle fibres. It is what happens when muscles contract. If you cut through a muscle, you will see it is covered in a layer of connective muscle tissue known as the epimysium. This protects the muscle from friction.
At the cross-section of the muscle there are bundles of fibres surrounded by connective tissue called perimysia. Each muscle fibre contains cylindrical organelles called myofibrils. Each myofibril can be broken down into segments called sarcomeres.
This is a model of part of a myofibril called a sarcomere. The myofibril contains actin and myosin filaments, and the sarcomere is the functional unit of the myofibril. Here the muscle cell is at rest. You can tell because the H zone is elongated due to reduced overlapping of the myosin and actin filaments. The H zone is the area containing only the myosin filament, and the actin is the thin filament seen in blue.
This is a sarcomere contracted. The model shows overlap between the actin and myosin. Because the thin actin filaments are closer together, there is a reduced potential for cross bridges to form. The sarcomere is shortened as the actin and myosin slide over each other. The Z lines are closer together and the H zone is covered. This happens in hundreds of sarcomeres, causing the muscles to contract. This is at rest. The tropomyosin and troponin are wrapped around the actin. The troponin active sites can't connect to ADP, so there is no binding site available.
Calcium has entered the cell through the sarcoplasmic reticulum. The brain sends signals to the motor neuron which generates actin potential. The active sites move so ADP can connect. Troponin becomes available to the myosin head. The myosin head connects to the active site, creating a cross bridge. During the power stroke, energy is released by ADP. It attaches to the myosin head and then is ejected off. This creates energy.
ATP then re-enters the muscle cell and attaches to the myosin head. ATP is then hydrolyzed, which transforms it to the ADP. This releases the myosin head off the actin. In order for contractions to continue, calcium and ATP must be present in the muscle cell.